All right, guys. So what I want to do is just take this opportunity to recognize the accomplishments of the students in the program. One of the things I did was to write to the professors and ask, do you have students in your lab who have won prizes and awards in the past year? And I was really impressed both by the quality, the number of awards that the IPN students have been bringing in, but also by the enthusiasm of the professors. So everybody wanted to brag about their students, which was kind of nice. So what I have for you is a partial list. So if you're not on the list, please accept my apologies. I'm starting with some of the awards that are outside of the kind of classic mainstream federal and provincial, and then I'll work my way there, and then finally come to the awards that will be presented today. So I received a number of emails from labs throughout the university, and as you can see, there are awards ranging from this National Research Training Platform in Neurodevelopmental Disorders, CREATE and MyTax Awards for the Cadre Lab, and the Bashevan Lab. Motare received the Unique Excellence Scholarship. Adele Rabovska from Van Mile Lab received the PEO, the International Peace Scholarship Award. In Madeline Sharp's lab, a number of students received CRBLM Travel Awards. Austin Milnerwood's lab, Anusha received the MacArthur Travel Award. In John Sakata's lab, Xinghao Yunwan received a MyTax Global Link Award. Maya Getty's lab, we have Larissa and Jing Tian, who received the McBain Scholarship and the McGill China Studentship. Massimo Avali's lab, Xian Wang, received a studentship from the Savoy Foundation. Keith Marai's lab, Sylvie LaHaye, received a fellowship from the Transforming Autism Care Consortium. Sylvain Baillet's lab, Ellie Hill, received a Transitional Research Prize and the 12th Annual Bruce Williams Pediatric Special Research Day Prize. And Jason De Silva Castanera received a CIHR Brain Star Award. So many great prizes. From the Diaz Lab, Jamie Beaulieu received the Stem Cell Network and MyTax Industry-Based Industrial Partnership. From the Flores Lab, Jose Restrepo received the Layton Addiction Award, Research Award, and the Douglas Presentation Award. Whoops, sorry. Shama Hook, the Fulbright Research Award. Tanya Capolicho received the Marie Giguère Travel Award. From Naguib Meshawar's lab, Claudia Beliveau received the Stratas Foundation, Brett Widerhold Scholarship, and Rithilwe Mpai received the Botswana Government Top Achiever PhD Scholarship. From the Bernard Lab, Niti Jain and Laura Lentini received MUHC Research Institute Fellowships. And from Ted Fahn's lab, Ghislaine and Zaid received Parkinson Canada 2-year graduate student awards. This is just a partial list. It's the ones that were sent to me by supervisors on request, and obviously congratulations are due. In addition, there are a number of formal awards that are given out by the Faculty of Medicine, HBHL, and others. So we have a number of recipients of the FacMed Faculty of Medicine Award at the master's level. And I won't read all the names, but I want to make sure that everybody's recognized. And as well, a large number of Faculty of Medicine PhD awards. So by far, IPN is doing extremely well in these competitions. HBHL, we've got a large number of master's and doctoral awards. So congratulations to the students here for receiving these very competitive and quite impressive awards. And then at the IPN Internal Awards competition, which I played a role in running this year, oops, it's been changed in size on the screen. We have many students who were recipients of the internal awards. There are also a number of awards that are issued from the Montreal Neurological Institute, so the Jean Timmins Costello Award, the Molson Neuroengineering Award, and then a couple of top-up awards for outstanding students, the Chippendale Award and the CNANS Common Award. And so congratulations to all the students who received these internal awards. For external awards, we did extremely well this year in the FQRNT and FRQS competitions, especially at the doctorate level. So for the FQRNT master's and doctorate level, you can see we have a number of awards here. Congratulations if your name is on the list. And for the FRQS master's and doctorate awards, again, many, many students were fortunate recipients. And so this is a great job. The FRQS award is one of the few awards that's actually available to international students as well. And so because of the strength of our student body, I think we do very well in this competition in particular. And then finally, for the Canadian federal level awards, we have done extremely well this past year. At the master's level, CIHR, CGSM, the following students received awards. And at the NSERC CGSM level, again, more awards were received. So fantastic job, everyone. The CIHR, CGSD level. These are really outstanding level awards from the Canadian Institutes of Health Research, from SSHRC, and from NSERC. So, great job, guys. I wanted to just put all the names up because I think more than anything else, it's impressive to see the number of students in our program who are doing really well recognized work. And so congratulations to all of you. In addition, so we have a number of awards that are really sort of high-level specialty awards for students in the program. The Tomlinson Award is essentially one of the highest internal awards that's offered by the university. And this year, Mingyu Wan received the Tomlinson Award from IPN. The Desjardins Award is one which is issued by the Montreal Neurological Institute, essentially for the top PhD student in the annual competition. And Hyo Lee was the recipient of this award this year. So congratulations, Hyo. There are also 4 students in the program who received Vanier CGS awards. And although the results are still under embargo, so I can't name names, the number 4 is quite impressive when we consider that there are only about 120 of these awards given out nationwide. So a good fraction of them went to IPN students this year. Okay, so for the award session today, and again, I apologize for the formatting shift when switching computers, but the last 2 awards that I'd like to talk about are specific for the rotation for the retreat. The first is the IPN Excellence Award. This year's recipient is Calvin Wong. And I again apologize for the formatting. Calvin is a student in the Kotorsky Lab. And the nature of the IPN Excellence Award and the Anne and Richard Sievers Award are that we ask the supervisors throughout the university, throughout the program, is there any student who's nearing the end of their studies, who's doing really extraordinary work that would benefit from having a little bit of an additional funding, a little bit of additional time to take their project from what is already superb and push it to the next level. And so each year then, we do a competition, we evaluate the students' submissions, and this year we had 2 very, very close finalists. The first place winner is the Anne and Richard Sievers Award winner, Laura Suarez. So the Anne and Richard Sievers Award is an award which was essentially the result of a gift from Mark Sievers, who is here today to be able to issue the award. And we're very lucky to have him here. Maybe, Mark, would you mind coming up or coming up with Laura and also with Calvin? So I don't know if you want to get on the podium or
no, we'll just
stand here. Okay. So I just want to acknowledge Mark has been an extremely generous contributor to the IPN program. This is the 12th year of the Anne and Richard Sievers Award. And over the years, it supported really outstanding work. 2 years ago, we were lucky enough to have had a 10th anniversary reunion of students who have received the Sievers Award in the past. And it was truly an impressive event because the students who have received that award have almost all gone on to do extraordinary work and have developed their careers to an extreme and impressive level. And so we expect that the recipients of this year's award will also follow that same trajectory. So Calvin, as the runner-up for the award, the IPN makes a special award for you, which is the IPN Excellence Award. And so I'll just congratulate you first. And Laura Suarez, who is in the Message Lab, her proposal was just found to be innovative, really kind of publications up to now have been very high quality. And I think people really believe that with this extra bit of time and push, that it's going to be a truly impressive PhD. So thanks to you, and thanks to Mark for this opportunity.
Thank you. You're more than welcome.
Thank you so much. So what we like to do is give the two award recipients a chance to present their research. So there'll be a brief 10-minute talk from Calvin about his research in the Kotorsky lab, followed by a 20-minute talk by Laura presenting her work. And I hope you all enjoy. So Calvin, I think I've got your slides here. Yeah, sorry. And I hope your slides didn't get disorganized the way mine did here. Okay.
Yep, looks fine.
Great. Calvin.
Thanks, Ed. So today I'll just give a brief talk of sort of my PhD thesis, what I'm working on. It's titled Mammalian Target of Rapamycin Complex 2, or mTORC2 Mediated Structural Plasticity in Chronic Pain. So I think I'll just touch on a little bit about chronic pain, why it's important. So it's a very ubiquitous and debilitating sort of condition that affects approximately 1/3 of the world's population. And it's also important because the current available treatments for chronic pain are mostly for symptomatic relief, and it doesn't really treat sort of the underlying mechanisms that are involved in the development as well as the maintenance of chronic pain. So it sort of drives research towards looking at sort of the fundamental basic questions as to how chronic pain develops and is maintained. And generally, chronic pain can be present in many sort of different modalities and ways. Generally, two sort of classifications are like inflammatory pain, which is characterized by tissue damage, and neuropathic pain, which is induced by direct injury to the nerve. So I'll just briefly go through the mammalian target of rapamycin complex. It's a highly evolutionarily conserved serine 3-adenine kinase, and it's very important in most cellular functions ranging from growth and survival as well as autophagy. And it presents in two functionally and structurally distinct complexes. The first is very well known in cancer research, particularly the first complex for its role in regulating mRNA translation and protein synthesis. Much less is known about the secondary complex, particularly in the field of chronic pain. And that's sort of the foundation of my sort of PhD research. So I'm looking at how mTORC2 might be involved in mediating this development and maintenance of chronic pain. What is known is that it regulates structural plasticity through downstream regulation of downstream signaling to regulate actin dynamics and polymerization. And importantly, it also phosphorylates this AKT kinase at serine 473. So this acts as a direct readout for looking at mTORC2 activity, as it can't be directly measured itself. So what our lab has noticed, as well as many other labs for a long time, is that there's an increase in this phosphorylated AKT at serine 473, so representing increase in mTORC2 activity, particularly in nociceptor neurons in the dorsal root ganglia after inflammation. We show that after 6 and 24 hours of inflammation that there's robust increase in phosphorylated AKT in these nociceptors. So what you see here is that we use TD tomato reporter mice, and we use it to identify NaV1.8 positive C fiber nociceptors in the dorsal root ganglia. So there's an increase in mTORC2 activity, particularly in these cells. And for a long time, it's been unknown what the functional significance of this increase is. So that's sort of my sort of research looking at that. To do this, we generate transgenic mice where we impair mTORC2 activity specifically in these nociceptive neurons that receive pain input. So how we do this is that we use the Cre-LoxP system, and we delete specifically Richter, and it's the main protein that characterizes mTOR complex 2. So by deleting this protein, we can impair mTORC2 activity, specifically in NaV1.8 positive C fiber nociceptors in the dorsal root ganglia. And we just confirmed with Western blot that both Richter as well as phosphorylated AKT, serine 473 protein levels are reduced. So it sort of confirms that we correctly knocked out and impaired mTORC2 activity in these nociceptive neurons. When we subject these transgenic animals with conditional deletion of Richter and resulting mTORC2 impairment in nociceptive neurons in the dorsal root ganglia, we see that it alleviates inflammatory pain when we use two tests. First is the von Frey test on the far right, looking at mechanical sensitivity, and the second test, the radiant heat pulse withdrawal test, looking at thermal sensitivity. In both tests, these Richter conditional knockout animals show greater alleviation in pain. So they have higher thresholds, resulting in less sort of sensitivity of pain. We also use an innovative sort of assessment looking at facial features to assess spontaneous pain. Involves looking at 5 facial features that are distinct for this spontaneous or grimacing sort of pain. And we also show that these conditional knockout animals show alleviation of following inflammation insult as well. So I've shown up to this point that mTORC2 activity seems to be increased in conditions of inflammation, and when we impair its activity, it sort of rescues this sort of pain. So we wondered whether activation of mTORC2 directly would promote sort of pain-like behavior by itself. To do this, we administer a small molecule activator, a pharmacological activator that is specific for mTORC2, and we show again, looking at mechanical and thermal sensation, that this activator promotes pain compared to animals that receive vehicle control, as well as these animals show more sort of grimacing features that are pain-like. So we're focusing specifically on nociceptive cells that are present only in the dorsal root ganglia of the peripheral nervous system. These cells send their projections to the epidermis, where they sense stimuli, and they relay this information to the spinal cord, where it ultimately is processed in the brain. So we thought to first look at sort of these processes. Another reason why is because mTORC2 is, again, related to structural plasticity, so it made sense sort of to look at the processes, whether it be in the epidermis or the spinal cord. We just first chose to look at the skin. And what we noticed was that when we induced inflammation, local inflammation in these animals, this is, again, animals that are TD tomato fluorescent reporter mice, identifying NaV1.8 positive nociceptors and their processes at the level of the epidermis. And we utilize high-resolution air scan confocal microscopy to show that there's this hyperinnervation and growth following inflammation insults at day 1 and 3, post-day after inflammation. But looking at these Richter conditional knockout mice with mTORC2 activity impaired, it sort of inhibits this growth and plasticity that we see in mice that have normal mTORC2 functioning. We also show that again with our small molecule pharmacological activator, that when we administer it promotes this hyperinnervation and growth at the level of the epidermis. and this is also rescued in the Richter conditional knockout mice. So I sort of show up to this point that mTORC2 plays a role in structural plasticity that is relevant for chronic pain, but we wanted to rule out whether mTORC2 activation of it has a direct effect on intrinsic excitability of these nociceptors. So what we did was that we performed two experiments to measure parameters of intrinsic neuronal excitability. What we did here just briefly was that we took neurons from the dorsal root ganglia and we cultured them. We applied this activator for mTORC2, this pharmacological activator, and we saw no changes in various parameters. And we also injected the pharmacological agent as well in vivo into animals, and then took neurons and cultured them and recorded from them. We also applied a very highly innovative technique where we did in vivo calcium imaging of these neurons in the live mouse. So under anesthesia, we measured calcium transients, and we saw that administration of this pharmacological activator for mTORC2 had no effect as well on excitability. We also utilized a computational modeling approach that sort of supports our hypothesis that structural plasticity at the level of the epidermis is sufficient enough to sort of produce increased neuronal excitability. So what you see here is that it's a schematic of the peripheral nociceptive tree here, where at the branches innervate the skin, and then it goes to this sort of cylindrical shape representing the cell bodies, the dorsal root ganglia, and then we measure at the level of the central terminals. And when we applied our model, it seems to this growth and innervation that we see in our stainings is sufficient to produce increased number of action potentials, resulting in increased excitability. Lastly, we just want to cap off with testing a therapeutic approach, because there are no pharmacological inhibitors or any other form of inhibitors for mTORC2 and Richter. So we opted to use antisense oligonucleotides, and we show that it reduces both Richter protein as well as phosphorylated AKT protein, and it sort of rescued this hyperinnervation that's induced by inflammation as well as pain-like behavior. So just to conclude, under the normal condition, you have these neurons that sense pain, and they send their projections out to the epidermis in the periphery, and you have a normal sort of sensation of this noxious stimuli, and it results in action potential firing. But following an inflammatory context that activates mTORC2, it results in this hyperinnervation and growth of peripheral terminals at the level of the skin, and it results in an increased amount of action potential firing that's associated with this inflammatory hyperalgesia. So I just want to thank everyone in my lab, as well as my supervisor and all my collaborators involved, and the funding bodies.
Any questions for Calvin?
Okay. Good afternoon, everyone. Thank you very much for being here. For me, it's a real pleasure and an honor to be giving this talk today at the IPN Retreat. I want to start by saying thank you to Mark Sievers for this amazing opportunity. So without any further ado, let's begin. So the title of my PhD research is Reverse Engineering and Reengineering the Brain. So ever since I started to pursue an academic career, my research interests have been at the intersection of these three fields: artificial intelligence, engineering, and neuroscience. To better understand what I'm working on right now, I thought it would be worth to tell you a brief story of what I've done in the past. So in the past, the focus of my research was at the intersection of artificial intelligence and engineering. So this is how the story goes. So nowadays, modern computers allow us to do incredible things. We can browse the internet, we can perform advanced scientific simulations. They enable life-saving medical imaging. They have allowed us to explore the universe. They support financial and global communication networks, but most importantly, computers allow us to share memes on social media. So with no doubt, computers have revolutionized society. But what if we wanted to build a computer that could hear and see and perceive the world as we humans do, or a robot with the agility of a dog or a cat? And this is, of course, the goal of artificial intelligence. Now, as far as we can tell, we are our brains. All of our thoughts, memories, motivations, and personalities are encoded somehow up there. Now, from an engineering standpoint, one way to better understand biological brains is by trying to build one. And if we could build such artificial brain, what would it tell us about ourselves? So that's the question that inspired me and has inspired many to become researchers in the field of AI. Now, in the past 5 years or so, artificial intelligence has made incredible progress in trying to replicate human-like intelligence. So I'm sure you have heard of self-driving cars, or you have noticed that language translation has improved dramatically. Or maybe you've noticed that now you can talk to your smartphones, or your smartphones can even recognize your face in order to grant you access to the different apps. Now, one of the most impressive things from my personal point of view that AI researchers have achieved in the last couple of years is the ability to replicate a creative type of intelligence. It's such that now you could have your own art gallery entirely conceived by an AI. And this AI would be DALL-E, not exactly Salvador Dalí, but I'm talking about DALL-E, the latest OpenAI generative algorithm. All of these things have been possible thanks to breakthrough advancements in the science of artificial neural networks. Now, artificial neural networks are algorithms that were initially designed to help us better understand how the neurons and their connections or synapses in our own brains work. These are thought to be massively parallel in the sense that they can perform multiple operations at the same time. Unfortunately, our current computing architectures do not work in this way. They perform one operation after the other. Furthermore, every operation in current computers requires information to travel back and forth between the memory and the processing units, and this translates into high computational and energy costs. So because of these reasons, modern computing architectures are not ideal to simulate these artificial neural networks. However, despite this, artificial neural networks have become the state of the art in many fields. So just to give you an idea, our largest and most powerful network is only a tiny fraction of even a mouse brain. And for instance, for simple, or not simple, but tasks like speech or video recognition, the size of these networks are of about 1 million neurons. And even for these comparatively small networks, training can take up months, without mentioning the amount of energy that the training requires. So if we want to build truly scalable artificial neural networks, we need to fundamentally rethink our computing architectures. So we need to design processors that are massively parallel as biological networks are. So the field that tackles this problem is called neuromorphic engineering. So neuromorphic engineering moves beyond the standard computing paradigm in the sense that they try to build physical neurons and synapses circuits onto a chip, and in a way that they mimic biological networks. So here, for example, you have two photographs of these neuromorphic processors seen under a microscope. So they're impressively similar to what we would expect from biological networks. Now, unfortunately, these neuromorphic processors are not scalable either, but in a different sense. Whereas conventional processors take up too much time and energy to simulate these neural networks, neuromorphic processors take up too much space. So as you try to pack more and more neurons into these chips, the amount of wiring that is required to connect all of them really or quickly gets out of hand, even for relatively small networks. Fortunately, biological networks share the same constraint. Now, in both neuromorphic networks and biological brains, we need to connect a large number of neurons with modifiable synapses in a scalable way. The advantage here is that biological networks have actually found a solution for this. And this is exactly the question that I'm trying to answer during my PhD. How are neural networks connected? Now, fortunately, we're in the big data and the omics era. So thanks to advances in neuroimaging technologies, such as diffusion-weighted MRI and reconstruction methods, such as tractography algorithms, we can now map the wiring patterns of biological networks in such a way that we can reconstruct the structural or the anatomical networks of biological brains. These structural networks are also known as connectomes. Okay, so now I'm going to talk about what I've been doing for my PhD studies. So my research now is more focused at the intersection of artificial intelligence and neuroscience. So my PhD project revolves around the question of the link between structure and function in biological neural networks. So as you may know, the link between structure and function is an ubiquitous concept in biological systems across all spatial scales, from molecules like proteins, cells, up to the biomechanics of animals' skeletons. So how do we see this link between structure and function in the brain? So the brain can be modeled as a network of functionally specialized brain regions that are physically connected by anatomical links. We refer to this network of anatomical connections as the structural connectivity network of the brain. And this structural network forms the scaffold on which information propagates through the brain, and therefore it plays an important role in shaping how neural dynamics unfold over time. Now, functionally specialized brain regions not only interact physically, but also functionally through coherent dynamics. And we capture those functional interactions via what we call functional connectivity networks. Now, the co-activation patterns that emerge from those functional interactions are thought to be the neural processes that support cognition and behavior. But how do structure, dynamics, and function interact to support the computations that underlie cognition and behavior still remains one of the biggest questions in neuroscience, and my PhD project is directed towards answering that question. So specifically, what we've been doing is we combine MRI connectomics and reservoir computing to investigate the link between macroscale connectivity and the computational properties that emerge from network dynamics in the human connectome. So by MRI connectomics, I refer to the process of reconstructing the human connectome from MRI data. And reservoir computing is the computing paradigm that we're using to test the computational properties of these biological networks. Now, reservoir computing, in a very broad sense, is this idea that we can use dynamical systems to perform computations. Now, in its simplest architecture, reservoir computing consists of an input layer. Then we have a reservoir, which is a recurrent neural network. And then we have a readout unit, which is typically a linear model. Now, in contrast to traditional recurrent artificial neural networks, we do not train the connections within the reservoir. The only connections that we train are the ones that go from the reservoir to the readout unit, which correspond to the coefficients of the linear model. So you might be wondering, if we don't train these connections, how does reservoir computing work? So the idea is that we have an input signal. We introduce that signal to the reservoir through the input layer. Then the signal propagates, and then we're going to read the activation states of some of the units in the reservoir, and we're going to use those activation states to train the linear module to perform a particular operation. Now, the reason why it works is because the reservoir is so nonlinear. It's nonlinear that it performs the nonlinear projection that the reservoir performs into this space of higher dimensions is supposed to transform any nonlinearly separable problem into a linearly separable one. So as you might imagine, because the reservoir can take any network architecture, we use the human connectome to constrain the connections within the reservoir, and then we use these connectome-informed reservoirs to perform a memory task. Now, in this memory task, we use a uniformly distributed input signal. We introduce that signal into the reservoir. The signal propagates through the network, and then we're going to read the activation states of some of the output nodes. We're going to use those reservoir states to train the linear model, in this case to perform an approximation of a delay version of the input signal, where tau here represents the delay. We do this for different values of the delay, and for every value of the delay, we obtain a performance that is a correlation between the target and the prediction. Because we wanted to obtain a memory, like a single memory capacity estimate for the system, what we did is that we summed those correlations across delays. We were also interested in seeing how the dynamics affect performance. So for this, we use a parameter alpha with which we scale the connectivity, the coupling strength of the connectivity matrix. And by varying the value of alpha, we can transition the dynamics of the reservoir from stable to chaotics. So for different values of alpha, we perform the exact same procedure such that at the end, we end up with a performance curve of memory capacity as a function of alpha. Alpha values that are lower than 1 correspond to stable dynamics. Alpha values close to 1 correspond to critical dynamics. And alpha values that are greater than 1 correspond to chaotic dynamics. Now, for the input nodes, we use the subcortical regions of the network. And for the output nodes, we use the concept of intrinsic networks. So intrinsic networks are a functional connectivity-based partition of the cortex into regions that co-activate during tasks and that tend to share a common function. So for this work, we use these 7 networks partition. So using these 7 networks, we perform this memory task. So for each of them, we obtain a performance curve of memory capacity as a function of alpha. This is just a sanity check. We know from dynamical systems theory that the memory capacity of a system tends to be enhanced by stable dynamics, and as the dynamics transition to chaotic, the memory capacity of the system tends to decay. Now, the first thing that we did was to look at the memory capacity of the human connectome as a whole. So for that, we average across intrinsic networks, and this is the distribution across different connectomes for the memory capacity as a function of alpha. So again, this is consistent with what we saw in the previous slide. Next, we asked 2 questions. The first one was whether the memory capacity of the human connectome depends on the underlying network topology. So for that, we built a family of reward networks by randomly swapping edges in the network such that we preserve some local properties. And then the second question that we ask is whether the partition of the connectome into these 7 intrinsic networks provides any computational advantage. So for that, we use a label permutation model in which we randomly permute the labels of these intrinsic networks. And then using these 2 family of null models, we perform the exact same memory task, and then we compare that against our empirical networks. Now, the important observation here is that the human connectome outperforms specifically these 2 null models at the critical point, at that transition between stable and chaotic dynamics. And so basically, these results are showing that the computational advantages granted by the unique topological features of the human connectome, as well as by its modular organization into these 7 intrinsic networks, are optimized specifically in the context of critical dynamics. And this result is interesting because it kind of supports this old hypothesis that biological brains operate at that critical point, which is a dynamical regime that is thought to confer optimal information processing properties to the network. Now, here, I'm just showing again the result of comparing the human connectome against the reward networks. Now, the thing is that these reward networks do not take into account that the brain is a spatially embedded network. So also, the brain as a biological system, it has to satisfy certain physical demands in the presence of limited resources. So, for instance, one of those demands is the growth and maintenance of axonal projections, which entails certain material and metabolic costs. And these costs increase with the length of interareal connections. So if we look at the connection length distribution in the human connectome, we can see that there is a prevalence for short-range connections. And if we compare that distribution with the distribution of the reward nulls, we can see that this distribution shifts towards the right, which means that there is a higher proportion of long-range connections. And this, again, translates into higher metabolic and material costs. So if we take into account those costs due to wiring, and we estimate memory capacity per wiring cost, we can actually see that the human connectome outperforms these randomly rewired networks regardless of the dynamical regime. And these results support the idea that biological networks are economical networks in the sense that they optimize computational capacity while minimizing wiring costs. So there are more results from this first set of experiments. So if you're interested, this research was recently published in the journal Nature Machine Intelligence. So if you're curious, I would invite you to check it out. Now, because we believe that this hybrid approach between neuroscience and artificial intelligence really offers an opportunity to explore the link between a structure and function, or between a structure and computational properties in biological networks, we decided to build a toolbox, a Python-based, an open-source Python-based toolbox, Contour-RES. It is currently available in GitHub. It's still under development. But basically what this toolbox allows researchers to do is to perform this general workflow. There are some inputs that come from the user. But probably the most interesting part is that we also added more cognitive tasks. So now we can explore other cognitive domains. These tasks come from a curated collection of neuroscience tasks designed for supervised learning in artificial neural networks. So it really gives an opportunity to explore that link between a structure and computational properties in other cognitive domains. Another application of this same framework is to study the link between a structure and function across phylogeny. So we have this data set of the connectomes of about 120 plus mammalian species. These are some examples of the connectomes that are included in that data set. So this will also allow us to explore how the link between structure and function has changed throughout with evolution. Okay, so I just want to briefly tell you how the results of this type of research could have potential applications in the long-term future. And those applications, of course, would combine the three fields. So basically, again, we're going back to neuromorphic computing or brain-inspired computing systems. So one of those brain-inspired computing systems that I've had the chance to work on before are called memristive networks. So this is a memristor, which is thought to be the fourth circuit element together with the resistor, the capacitor, and the inductor. And these memristors are thought to behave very similar to how synapses behave in our brain. So they are the ideal candidate to replace synapses in these neuromorphic processors. So I just want to compare how memristive networks compared to modern computing architectures. So as I said, these memristive networks, memory and processing, are one and the same process, very similar to how it occurs in biological brains. Whereas in modern computing architectures, memory and processing are different, completely different process, which actually translates into higher computational and energy costs. Also, these memristive networks are massively parallel, in contrast to the sequential processing that we have in current computing architectures. These memristive networks are also designed at the nanoscale versus the microscale of these chips of current processors. So if we understand how the brain is connected to produce certain computational properties, we can actually use that knowledge to maybe guide the design of these memristive networks and to say how memristors should be connected in such a way that we obtain a particular desirable behavior or computation. So with this, I want to thank to all the amazing members of the Network Neuroscience Lab. I want to thank my supervisor, Bratislav Mišić, I think, Guillaume Lajoie, my co-supervisor, the founding agencies, and last but not least, thank you to the Sievers family.
Are there mean, it's really beautiful work. And one of the questions is that I have the sense that although we're extremely efficient in terms of metabolic and energetic consumption versus computational output, it seems like that's also a limitation for biological systems, for brains. Do you think that it's inevitable then that a system could be built that isn't limited by those constraints that are limited to in a biological organism that would vastly outperform the biological system?
Do you think we are limited as biological systems?
I don't know. I guess that's what I'm asking. Yeah, are we limited because of the energetic and metabolic constraints imposed on us by biology?
I don't think we. That's a good point. I don't think that would be the only two factors that impose limitations on our capacities, I think. But maybe like building this type of neuromorphic processors could help us explore those, because we can kind of surpass those limitations in this hardware. So maybe it could be a way of exploring this type of questions. Yeah, it's very interesting.
Thanks. Any other questions?
Laura, explain a little bit more what happens in the chaotic states.
Could you repeat the question, please, Laura?
What happens in the
What happens in that right-hand part of the graph where what you describe as the chaotic states?
what happens in the brain, or what happens in that particular system?
Either in the simulation or in the brain itself.
Yeah, well, I can tell you in the simulation, at least. So the chaotic this regime is characterized by, as in like chaos in any dynamical system, is that it is very sensitive to initial conditions. So a very slight change in your initial conditions can result in very different results. But actually, having that property for some dynamical systems, depending on the type of task. So for example, if you are doing some sort of pattern recognition, you actually want your system to be able to produce different outputs when you have different inputs. So in that case, like chaos is beneficial. In the case of the memory capacity, as you see, the memory capacity was decaying, and that's because when you want a system to have memory, you actually require that it kind of settles at a stable point, because that's how you would characterize memory. I don't know, but yeah.
Okay. Thank you very much. Congratulations.
Thank you.
It's really innovative and creative work.
Thank you.
So I just want to emphasize that the Anna and Richard Sievers Award really is the most prestigious award that the IPN gives out each year, and so I really want to congratulate you on
Thank you.
what you've accomplished. The other thing, Laura was very smart to switch computers. She didn't get her fonts all screwed up, and so what I wanted to say was that in the course of getting a little bit flustered by that, I neglected to mention one very important thing. The IPN in the last year has instituted a systematic process of evaluating applications and grants through a standing committee. And this committee has done a remarkable job of processing a large number of applications this last year. And so I just want to let people know and extend thanks to the committee members, and in particular to the chairs of the committee. So Dominique Walker and Mindy Levin have been chairing the committee for the past year and doing a fantastic job, a huge amount of work, just based on the number of awards that IPN students have actually succeeded in winning in the past year. So I want to thank them for this fantastic job that they've been doing. Okay, so now we begin the next session. I will, Bratislav, if you want to go ahead and start. I'll allow you to introduce the session.
Yeah, of course. Hello, good afternoon. It's so nice to see so many people here. My name is Bratislav Misek, and I'll be chairing this afternoon's session on the structure, dynamics, and function in microscale brain networks. I really want to extend my thanks to IPN, to Ed, and everyone working with Ed for giving us an opportunity to represent some of the great brain imaging work that's taking place in IPN, and we're very excited to hear what this audience thinks of this work. So I just have a couple of slides just to kind of introduce the topic, and then we'll hand over to our fantastic speakers. The jumping off point, as you just saw from Laura's talk, is that we live in an exciting time where we can map, image, and trace the connection patterns in many different brains. We can do this in various model organisms, such as C. elegans or the mouse, but most exciting of all for us, we can do this in the living human brain. So what used to be done with painstaking manual dissection in post-mortem brains can now be done relatively automatically using techniques such as diffusion-weighted MRI. And we can use to reconstruct the fibers that interconnect different neuronal populations to one another. And we can distill this data down into these intricate circuit diagrams that we call connectomes that you just saw. The idea here is that by representing the brain as a network of nodes and edges and taking a systems level perspective, we can actually situate our data and our field with respect to other scientific disciplines. So we can borrow concepts, methods, occasionally even theories from the folks who work on genetic interaction networks, who work on language networks, transport networks, social networks, and so on. And it gives us a real lingua franca with which to describe and to quantify and to articulate the architectural features of the brain. This is just one example of kind of the sheer variety of questions that we can ask. So for instance, how does the brain change over time, over very short or very long time scales? How are the different components of the brain organized across spatial scales? So how do the different circuit elements connect with each other and give rise to larger and larger, more polyfunctional circuits? And one kind of emerging idea that I think will be touched upon by at least a few speakers today is this idea that we are increasingly moving away from this idea of just focusing on macroscale brain networks where we pretend all the nodes in the brain are the same, but we're paying attention increasingly to the microarchitecture and to the biological details of each of these neuronal populations, and we're annotating them with lots of different features that we can get from other tracing, imaging, and histological analyses. This is incidentally a figure from a review paper from our previous speaker, Laura Suarez. So that kind of gives you an idea of what we're going to be doing. We have 4 absolutely fantastic students from IPN who are going to touch on various questions related to both brain structure and function. How does this relate to microarchitecture, and also how does it relate to various diseases? So I would like to, I don't know if, I haven't seen her yet. I don't know if Jessica Royer is here. There she is. Great, so I'd like to invite Jessica Royer to come up. Jessica works with Boris Bernhardt and Birgit Frauscher, and she'll tell us about her work on the role of microarchitecture and microscale network organization in epilepsy. So please join me in welcoming Jessica.
There you
go.
Awesome. Let's see. Sit up here. All right. Okay, perfect. All right. Well, good afternoon, everyone. My name is Jessica. I'm really pleased today to be presenting some of the work that I've been doing as part of my PhD here at the IPN. So today I'll be talking to you about temporal lobe epilepsy, specifically looking at changes in brain microstructure in these patients and how this relates to changes in the functional network organization, specifically of episodic memory networks. So just a few words on epilepsy to start off. So we know that this is temporal lobe epilepsy is the most prevalent type of drug-resistant focal epilepsy in adults and is characterized by changes in brain microstructure. So mesiotemporal structures are particularly affected in this condition. So disease-related microstructural damage is often visible in histological specimens taken from post-surgical tissue. So we can see often hippocampal sclerosis in these specimens, which manifests as variable degrees of cell loss and gliosis in the hippocampus. But these microstructural changes can also be seen in different MRI sequences, which has really opened the way to investigations of structure-function interactions in these patients. Just research, okay. So also, temporal lobe epilepsy has really emerged as this unique disease model to study the structural substrates of episodic memory, in part thanks to these new investigations that we can perform on these patients with MRI. So we know that TLE patients often present with cognitive impairment that can affect different cognitive domains, but episodic memory is often affected. And these deficits have been repeatedly associated with the atypical organization of memory circuits that involve the hippocampus, but also the degree of hippocampal damage indexed by regional volume reductions. But in addition to these very focal changes within the hippocampus, advances in structural imaging have also revealed more widespread structural changes in temporal lobe epilepsy. So patients often show these quite extensive and bilateral patterns of cortical thinning relative to healthy controls that can affect parietal, precentral, but also superior temporal cortices, in addition to the strong regional volume reductions that we see in the hippocampus ipsilateral to the seizure focus. And differences between patients and controls have also been seen in different diffusion imaging parameters, where alterations were found within the cortical sheet, but also in the superficial white matter, so near the boundary of the gray and the white matter. And these findings with diffusion imaging indicate some perturbations in the myelin sheath and axonal membrane integrity, as well as potentially increased extra-axonal space in regions that extend beyond the mesiotemporal lobe, particularly affecting more lateral temporal as well as lateral prefrontal and mesial prefrontal regions. And with high-resolution structural imaging, we can also now investigate the depth-specific nature of these changes across the cortex and study potential changes in regional laminar organization beyond the affected temporal lobe. So for example, in this study that used quantitative T1 imaging, which is a sequence thought to be sensitive to intracortical myeloarchitecture, the authors could observe significant signal alterations within the mesiotemporal lobe, but also in more extended structures like lateral temporal cortices and orbitofrontal regions. But at more superficial cortical depths, like we can see in these top figures here, signal changes were even observed in more occipital, parietal, and frontal regions. So overall, these studies using these more microstructurally sensitive sequences, but also diffusion imaging and structural imaging support this view of TLE as a more macro-scale system-level disorder in which a temporal limbic disease epicenter could be at the root of these more widespread structural changes in these patients. However, these structural changes might be depth-specific and also seem to be heterogeneous across regions, and it still remains unclear how memory function is related to these more distributed patterns of microstructural damage in TLE. And one approach that could allow us to capture these large-scale and regionally heterogeneous patterns of microstructural changes in TLE is by considering interregional patterns of similarity in microstructural properties across the cortex and how these patterns might be altered in the condition. And this macroscale organization of cortical microstructure has already been investigated in healthy populations, but also in different animal studies, and have been shown to follow this quite reproducible and distinct pattern. So we see this gradient of cortical microstructure that distinguishes regions according to the similarity of their intracortical microstructural characteristics, with similar colors in these figures representing regions that share similar microstructural properties. And we can see if we follow this gradient along this color axis that this gradient also tracks changes in different categories or subtypes of tissue in terms of laminar differentiation across the cortex. And importantly, we can obtain a similar gradient using myelin-sensitive imaging contrast like quantitative T1 imaging, which enables these in vivo depictions of this microstructural gradient in healthy populations, but also in different patient populations. So with this approach, we can now perform these subject-specific assessments of cortical microstructure that are not limited to local regions, but that are able to inform us on a region's more large-scale embedding within the broader microstructural landscape of the cortex. And we can then use these individualized gradients to anchor other features of interest, like functional network changes or different behavioral measures. So the objective of the study that I'll talk to you about today is really to see how TLE affects the organization of this large-scale gradient of microstructure and how these changes might relate to memory function and the organization of episodic memory circuits. So for this purpose, we studied 21 patients with temporal lobe epilepsy and compared them to 35 age and sex-matched healthy controls. And all participants underwent the same multimodal imaging protocol at a field strength of 3 Tesla, which included T1-weighted imaging, which allowed us to derive these cortical surface segmentations, which we then used to perform quantitative profiling of intracortical myeloarchitecture from quantitative T1 imaging sequences. And all participants also completed an fMRI episodic memory task, which I'll describe a little bit later in this talk. So in order to derive these subject-specific microstructural gradients, so we start by constructing intensity profiles that sample quantitative T1 image intensities within the cortex from the peel to the white matter boundary in the direction of cortical columns. And these intensity profiles are built for 10,000 unique points across the cortical sheet, which I'll also refer to as vertices. So these profiles showed variable shapes across different regions. So I show this here by comparing 2 regions, so primary visual cortex in blue and anterior cingulate cortex in green. So we can see that these 2 profiles are not only kind of shifted in terms of their mean intensity, but also show different overall shapes, which is consistent with what we know about these 2 regions, that they're very different in terms of their laminar architecture. And following this, these vertex-wise intracortical intensity profiles are then cross-correlated along the cortical sheet, resulting in these subject-specific matrices that represent the microstructural similarity of each vertex to every other across the cortex. And I'll just mention that all of this pre-processing up to the generation of these similarity matrices were performed with MikaPipe, which is an openly available pre-processing pipeline developed by our group. And we then applied diffusion map embedding, which is a nonlinear dimensionality reduction technique, to these similarity matrices to obtain eigenvectors or gradients of cortical microstructure in each TLE and healthy participant. For this study, we really focused on the first gradient, or the main axis of variance in microstructural similarity across the cortex, and compare these gradients across both groups, so healthy controls and TLE patients. So this first gradient of cortical microstructure, which is shown here for healthy controls at the top and TLE patients right on the bottom, followed a similar pattern in both groups by differentiating mainly unimodal sensory and motor regions, shown in these green-blue colors, from paralimbic and heteromodal cortices in pink and white. And although we see overall kind of a similar typical gradient pattern in both groups, if we plot the histogram of these values for each group, with TLE patients in orange and healthy controls in gray, we see that TLE patients tend to show this overall contraction of the microstructural gradient, particularly affecting the paralimbic anchor of the gradient. And we can directly compare these gradient patterns between TLE and healthy controls using surface-based linear models implemented here in the BrainStat toolbox. So if we directly compare these gradient scores between TLE and healthy controls, we see significant clusters of gradient reductions in TLE patients, specifically targeting more lateral temporal polar regions, as well as lateral prefrontal regions. So overall, these findings suggest a rather widespread loss of microstructural differentiation in TLE relative to healthy controls, with, however, more prominent effects seen in temporal and prefrontal regions ipsilateral to the seizure focus. So in other words, there seems to be this increased integration of microstructural gradient patterns across the cortex in TLE, particularly affecting more temporal and prefrontal regions. And to ensure that the results of this main analysis were robust and reproducible, we performed different control analyses. So we first repeated this case control comparison of the microstructural gradient while independently controlling for subject level measures of cortical thickness and blurring at the gray-white matter interface. And in both cases, we found that the results were robust to these common structural alterations seen in TLE. And we were also able to partially reproduce our main findings in an independent cohort of patients and controls that were scanned on a different MRI machine and at a slightly lower resolution. So specifically, we could well reproduce the reductions seen in the temporal lobe, but findings in the prefrontal cortex didn't survive correction for multiple comparisons. But overall, these kind of control analyses support that first, our findings were relatively specific to intracortical effects, and that the effects, at least seen in the temporal lobe, were robust in a distinct sample and less so in the prefrontal cortex. And next, to assess the specificity of our findings in microstructural gradients to temporal lobe epilepsy relative to other epilepsy syndromes, we turned to transcriptomics. So for this analysis, we cross-referenced findings from a recent epilepsy genome-wide association study with cortical gene expression data from the Allen Human Brain Atlas. And using fully preprocessed data that's aggregated in the Enigma toolbox, we could then obtain average gene expression maps associated with different epilepsy syndromes. And after controlling for the shared spatial autocorrelation between both features, we found that microstructural gradient contractions were significantly correlated with the average expression patterns of risk genes associated with temporal lobe epilepsy, but not with the patterns associated with other epilepsy syndromes, which suggests some specificity of the identified changes in microstructural organization to TLE-related genetic processes. And as part of this study, our participants also completed an episodic memory task while inside the MRI scanner, which allowed us to explore how microstructural gradient changes related to episodic memory network reorganizations and also recall performance in this task. So for this task, there was 2 phases. So first, an encoding phase where participants had to memorize pairs of images, 56 unique image pairs with half of pairs presented twice. And after about a 12-minute delay, patients completed the retrieval phase, which followed a 3 alternative forced choice design. So there was basically 3 images shown at the bottom of the screen, and the participant had to select which image was originally paired with the image shown at the top of the screen. So we first looked at functional connectivity during the encoding phase of this task by targeting regions of significant microstructural gradient contractions as seed regions, and that are indicated here by the little black markers. So this allowed us to explore how these regions that we showed had atypical embedding within the microstructural gradient space were interacting with other regions during memory encoding. And we found that both temporal polar and prefrontal seeds showed these distributed connectivity reductions to limbic and default mode regions during encoding. And these significant areas of connectivity reductions are outlined here in white. And we could also find that recall performance in the task was significantly correlated with the magnitude of microstructural gradient reductions in temporal regions here, but not with the gradient changes observed in prefrontal regions, indicating perhaps at least some behavioral specificity of the identified changes in microstructural gradients. So just to quickly wrap up, so we found that TLE patients presented an atypical cortical microstructural organization that extended beyond a mesotemporal disease epicenter towards more anterior temporal and lateral prefrontal cortices. So we know from prior work that either used in vivo imaging or postmortem specimens and post-surgical tissue that regional laminar architecture in TLE is often abnormal or atypical. And our approach builds on these findings by considering local alterations in laminar architecture within their kind of broader microstructural context, captured here by subject-specific depictions of microstructural similarity patterns across the cortex. So we can ask what might be going wrong in TLE to cause these more large-scale changes in cortical microstructure. So we know that the patterning of laminar subtypes in the adult neocortex is the result of these neuronal migration processes that are guided by different signaling molecules. So we know that in several epileptic disorders, there seems to be defects in this prenatal process. And notably in TLE, there seems to be a dysfunction of renin signaling pathways that have been associated with hippocampal sclerosis, but it's possible that dysfunctions in this pathway could also affect patterning of more extended cortical sites. Next, by combining this openly available transcriptomic dataset from the Allen Human Brain Atlas with results from a recent epilepsy GWAS study, we could also show that microstructural gradient contractions seen in TLE followed the spatial expression patterns of risk genes associated with hippocampal sclerosis. Transcriptomic mechanisms that support more kind of macro-scale brain organization are still quite an ongoing and important topic of investigation currently in neuroscience. So our results kind of offer some insight into this question by supporting a relatively specific spatial correlation between TLE risk gene expression and large-scale changes in cortical microstructure. And lastly, we demonstrate that regions with abnormal microstructural differentiation in TLE showed widespread disconnections to limbic and default mode subregions, particularly involving areas in lateral temporal but also ventral prefrontal regions. So we know that frontotemporal circuits involve really important regions for episodic memory function in humans, and perturbations in these interactions could impair encoding and the retrieval of newly learned material. And we show that microstructural changes in TLE is associated with the relative disconnection of regions within this network, which could possibly be impeding the efficacy of memory encoding in these patients. And in line with this idea, we show that indeed there seems to be this association between the magnitude of microstructural damage or microstructural changes in temporal regions and task performance in this episodic memory task. So collectively, our findings offer new evidence of perturbed structure-function interactions in TLE, affecting not only the mesiotemporal lobe, but also extending to more frontotemporal systems that support memory encoding. So with that, I'd like to thank all my lab mates, my supervisor, Boris Bernhardt, co-supervisor, Birgit Froscher, all the collaborators that offered valuable feedback for this work. And I'll also highlight the contribution of several openly available tools in completing this work. So we have different pre-processing pipelines, analysis tools for statistical analysis, and also an openly available data set. So I encourage you to check it out. So that's it. Thank you.
Wonderful. Thank you so much, Jessica. Do we have any questions? Get the ball rolling, maybe. Since you're correlating the intensity profiles across many depths, are you able to make inferences about what layers are affected in the disease?
So we maybe get closer. So we can't really. It's hard to directly relate the layers to the specific depths, because this might vary also across different cortical regions which have different thicknesses of different cortical layers. But this is something that maybe could be done in future work that can look at surgical specimens so once the patients undergo surgery to kind of remove the seizure focus, then there could be perhaps more targeted investigations for layer-dependent effects. Yeah.
We have no more questions. Please join me in thanking Jessica for a wonderful talk.
Thank you.
I believe our next speaker is Alyssa. Again, I haven't seen Alyssa here, but is she? There she is. Great. So next up, we have Alyssa Dye, who is a just very, very recently graduated student who worked with Malar Chakravarti and did, again, absolutely fantastic work. So we're very happy that we were able to snag her as she moves on. But yeah, so please join me in welcoming Alyssa.
Thanks so much. Hello, everyone. My name's Alyssa, and I'm really excited to be presenting my master's work today on mapping cortical morphometric patterns of the psychosis spectrum to symptom dimensions, brain function, and gene expression. So psychotic disorders such as schizophrenia are increasingly conceptualized as existing as clinical manifestations of phenotypes that exist along a continuum from health to illness. And in line with this continuum, hallmark features of these disorders, such as positive symptoms and negative symptoms, also exist at subclinical levels in the general population. And while these symptoms are often transient, they represent an extended psychosis phenotype, which we refer to here as psychosis spectrum symptoms. And importantly, these symptoms not only indicate higher risk for future psychotic disorders, but also risk of other threshold psychopathology in later life. And this kind of gives impetus to understanding the earliest emergence of these psychosis spectrum symptoms, which my work focuses on. Now, the psychosis spectrum is thought to capture kind of multiple interacting underlying susceptibilities related to elements such as neurodevelopment, neurobiological changes, clinical or behavioral manifestations, and also possible underlying molecular processes. And so an analytical framework that considers all of these different aspects is important for characterizing this phenotype. For one, psychosis spectrum symptoms are most prevalent in youth, concurrent with when the brain is in the crux of maturation, including active synaptic refinement, as shown here in this figure. And this is also in line with the theory that deviations from normative trajectories of development are what give rise to the elevated risk for diseases such as schizophrenia, which is also highest during this period. And so adolescence or youth in general provides a critical window to investigate psychosis spectrum symptoms because it allows us to identify neurobiological abnormalities at a time when therapeutic intervention might be able to take advantage of plastic change going on in the brain. And so to this end, magnetic resonance imaging, or MRI, offers the opportunity to characterize abnormal morphological characteristics through something called cortical surface-based analysis. And this essentially works by constructing these triangular meshes that represent the geometry of the peel and white matter surface of the brain, as shown here. And this allows for multiple morphological measurements to be computed at individual points across the cortex, which are represented as vertices along this mesh. And using this approach, we can extract multiple features of the cortex, such as cortical thickness, surface area, mean curvature, and local gyrification index, as shown here. And while cortical thickness and surface area are kind of more intuitive, mean curvature is a measure of folding that acts more on the millimeter scale level and reflects kind of the sharpness or the concavity or convexity of the curve of a surface, whereas local gyrification is more of a centimeter scale measure and reflects complexity in surface convolution. And these different features have differing maturational timelines and in different regions of the cortex, depending on sensory motor or association areas, and are also thought to reflect kind of interdependent but also distinct underlying biological processes. And so they can work together and give complementary information to a neurostructural signature of symptoms. And in line with that idea, psychosis spectrum symptoms have been shown to be associated with abnormalities in these features, such as reduced surface area and reduced local gyrification index, and this mirrors what we see in patient cohorts as well. But given that a lot of studies have focused on just a single measure of morphology, we were interested in whether psychosis spectrum neuroanatomy could be characterized by the coordinated variation of multiple morphometric features, which is a feature of brain development in general. And past work on a technique called morphometric similarity mapping has shown that by looking at covariance in metrics across the brain rather than a single one, we can actually identify more biologically meaningful networks. For example, Seelitz and colleagues showed that by correlating multiple different gray matter, curvature, etc. features across pairs of regions in the brain, we could identify these networks of areas with greater morphometric similarity that also share higher cytoarchitectonic and transcriptomic similarity, especially when compared to networks based on just a single feature, for example, cortical thickness. And while this specific technique is challenged by difficulties in recovering contributions of individual features to identified networks, more broadly it shows that integrating multiple metrics can allow us to potentially capture more meaningful clinically relevant differences in brain structure. From a neurobehavioral view, existing neuroimaging studies of the psychosis spectrum have often compared a single symptom domain or a summary measure of symptoms to brain measures, and this doesn't adequately capture the heterogeneity we see in symptoms. Instead, there's increasing evidence that the psychosis spectrum emerges from the variation of multiple distinct symptom dimensions, each of which represents a continuous phenotypic axis underlying multiple measurable symptoms along which a single individual can vary continuously. And we have 2 examples of such dimensions shown here in red and orange. And while a consensus model is still lacking, especially for youth, more broadly, these findings indicate that data-driven dimensions might better capture variation in neural abnormalities compared to something like case control approaches. And to further investigate how morphometric patterns in the psychosis spectrum might confer vulnerability for associated symptom patterns, we thought to integrate insights from cognitive function, from maps of cognitive function-related activation in the brain using this database called Neurosynth, which is a meta-analytic study of high-frequency cognitive or psychological related keywords in the functional MRI literature and associated activation coordinates in the brain. And we also sought to integrate kind of large-scale hierarchies of brain organization that have been found, again, from the functional MRI literature, and also to connect our morphometric patterns to gene expression, for example, of certain cell types, diseases, etc. And so our first aim was to link morphometric covariance patterns to data-driven dimensions of psychosis spectrum symptoms in youth. And for this, using the Philadelphia Neurodevelopmental Cohort, which is a large, non-help-seeking sample of youth that's also pretty racially diverse, we followed previous papers to derive a subsample of symptoms that are enriched for psychosis spectrum symptoms. And from these youth, we used structural T1-weighted neuroimaging scans, as well as symptom profile, psychosis spectrum symptom profiles, and sociodemographic features. And so to investigate patterns of variation in morphology in these psychosis spectrum youth, we extracted 4 metrics of interest listed here, so cortical thickness, surface area, mean curvature, and gyrification. And after regressing out kind of global measures to reduce their influence on these patterns that we find, we submitted these metrics to something called orthonormal projective non-negative matrix factorization. And this is a statistical technique that decomposes the inputs into 2 elements. So the first, in this case, being morphometric covariance patterns. So this describes cortical areas that in which metrics vary in a similar way across subjects, and also subject-specific loadings of the included metrics onto the identified morphometric covariance patterns, or MCPs for short. And the idea is that these 2 outputs are identified to maximally recapitulate the input, and the number of MCPs is chosen based on a split half stability analysis and also the gradient of the reconstruction error of the input. And the advantage of this approach is that it allows us to recover individual contributions of metrics to morphometric patterns, and also yields a non-overlapping and parts-based representation of the cortex that can be easily extended to other modalities to contextualize them. And we also ran an exploratory factor analysis on the behavioral side to identify major underlying symptom dimensions. So taking our 30 symptom items related to psychosis spectrum symptoms, we identified latent symptom factors that load onto the items, and then from there derived subject-level factor scores, which represent the expression of each latent symptom factor within an individual. And next, to relate subject-specific metric loadings onto each of our morphometric covariance patterns on the brain side, to subject scores on symptom factors on the behavioral side, we submitted them to a partial least squares analysis. And this essentially uses something called singular value decomposition to identify maximally covariant patterns of the brain and the behavior data in something we call a latent variable, and there are usually multiple of these that are found. And to evaluate the latent variables, we used permutation testing, so permuting the inputs a set number of times to identify which latent variables are significant. And this essentially generates a null distribution of singular values, which reflect the covariance explained in the data by a given latent variable. And to identify which brain variables contribute strongly and stably to a latent variable, we use bootstrap resampling, and this generates a null distribution of weights of individual brain variables. And to derive kind of like a normalized weight to identify strongly contributing brain variables, we calculated a bootstrap ratio, and this essentially is the ratio of the true weight of a brain variable to the bootstrapped standard error. So moving on to our results, we observed in NMF 8 morphometric covariance patterns in psychosis spectrum youth, and we show here the subject metric loadings onto each of our morphometric covariance patterns. And we basically see, given kind of the relative homogeneity in color, that the MCPs aren't driven by any single metric. And when we look at the patterns neuroanatomically, we see that the MCPs are largely bilateral and capture kind of contiguous brain areas that are also recognizable anatomically. For example, the lateral and medial prefrontal cortex or the precuneus. And in these maps, a darker color just indicates greater contribution of a vertex to a pattern. And on the symptom side, we identified 3 latent dimensions of psychosis spectrum symptoms. And these include negative and disorganized symptoms as our first dimension, including items such as lower occupational functioning, loss of motivation, etc. Our second dimension was disordered thoughts or sense of self, including items such as thought control and mind reading, paranoia. And then finally, our third dimension was hallucinations and abnormal perceptions, including kind of classical psychotic symptoms such as verbal and auditory abnormal perceptions. And from our partial least squares analysis, we actually identified 2 significant latent variables, but here we'll just focus on the second latent variable because it captured a relationship between our symptom dimensions and our MCPs, which was more relevant to our research question. And here on the left, we show the behavioral features of the latent variables, and on the right, we show in the grids the profile of morphometric changes within each of our MCPs that was associated with the behavioral pattern. And so in this latent variable, we see that older age, male sex, African American race, as well as higher severity on the disordered thoughts or sense of self dimension is associated with brain changes, including reduced gyrification index in MCPs 1 and 4, both increased and decreased cortical thickness in MCPs 1 and 3, decreased mean curvature in MCP 2, which is kind of the visual area, and decreased surface area in MCP 6. And given their contribution to this pattern, we refer to these contributing MCPs as those linked to disordered thoughts or sense of self in the coming slides. So our second aim was to link our identified MCPs to brain function. And to do this, we first identified their spatial correlation with meta-analytical activation patterns of certain cognitive terms derived from neurosynth. So here we have an example shown for the reasoning term. And this analysis showed that MCPs broadly correlated with distinct cognitive domains, of which the highest correlated terms are shown in the word clouds, and I've kind of summarized them on the sides. And what's interesting is that if we focus on those MCPs that we linked just now to disordered thoughts or sense of self, they are generally more correlated with kind of higher level processes, for example, executive control and emotional experience. And we secondly sought to link our MCPs to what are called functional connectivity gradients. And this follows from converging evidence that there are these kind of large-scale patterns of organization in the brain, particularly differentiating unimodal and transmodal areas. So unimodal areas in this case refer to more primary sensory areas in blue, and more transmodal areas are kind of more association cortex. And so we sought to situate each of our MCPs spatially along maturational expressions of this gradient given the age of our sample. And so we first show the results for the adolescent gradient, which matched our sample the closest. And we show in this plot distributions of gradient values that were extracted for each component from this adolescent gradient map. This is adolescent gradient 1, so the most dominant gradient in adolescence. And if we focus again on MCPs that were linked to disordered thoughts or sense of self, we see that MCPs 3 and 4, as well as MCPs 6 and 1, are kind of localized more towards the unimodal and transmodal ends of the gradient, respectively. So here, more negative values indicate proximity to unimodal, more positive values indicate proximity to the transmodal end. And in this slide, we integrated results from the child and adult gradient maps as well to kind of look at maturational trajectories of our MCPs along these gradients. And we summarize kind of the change in the relative rank of individual MCPs from the unimodal to transmodal side in this line plot. So sharper turns and greater magnitude of turns indicates greater change in their position along the gradient. And we see again, if we examine those MCPs that were related to the psychosis spectrum symptoms, which are indicated by crosses in the violin plots and dashed lines in the line plots, it appears that MCPs that are initially more unimodal, such as 2 and 3, are the ones that kind of shift most dramatically across adolescence and adulthood, whereas those that are more transmodal, such as 6 and 1, have relatively stable positions across the 3, but still show kind of noticeable changes in the distribution, which indicates some maybe more subtle functional reorganization. And finally, we asked whether specific cell types, biological processes, or diseases might underlie the MCPs that we've indicated might reflect a macrostructural vulnerability signature in this sample. And to do this, we leveraged data from the Allen Human Brain Atlas, and specifically, we used it to extract tissue samples taken from postmortem brains that were kind of spatially within the region of each of our MCPs. And each of those samples had a profile of gene expression collected. So we inputted the sample level expression data into a matrix that was grouped by the MCPs along Y, and then that in turn was submitted to another partial least squares analysis. And this variant essentially identifies differences between groupings of variables in a single data set. So in this case, the groups are the MCPs, the morphometric covariance patterns, and the observations are the individual tissue samples. And in this case, this analysis identifies patterns of gene expression reflected in the gene weights that maximally differentiate our input groups, so our MCPs. And by thresholding the gene weights, we can identify a PLS positive and a PLS negative gene set that characterize different MCPs, and we can submit that to enrichment analyses. So we present here the significant latent variable from this analysis, and in this plot, MCPs that are differentiable from each other are shown by opposing colors, so red and blue, and the magnitude of the contrast value indicates essentially their degree of contribution to the contrast, so how much their associated gene pattern contributes to this pattern of differentiation. And the contrast values are visualized on the brain plot as well. And based on this interpretation, we see that PLS-positive genes, which are represented by red, primarily characterize MCPs 4 and 6, and PLS-negative genes primarily characterize MCP2. So this latent variable essentially mostly differentiates MCPs 4 and 6 from MCP2. And to better understand the gene sets themselves, we can look at their cell type enrichments to start. And here we calculated an enrichment ratio using genes from 7 canonical cell classes. So we looked at the ratio of genes from the PLS positive and negative gene set that were preferentially expressed in each cell class. And in this box dot plot, the colored dots indicate the true enrichment ratio for a cell type. The box plots are the null enrichment ratios from 10,000 random gene lists. And the direction of difference between the two indicates whether that cell type is over or under enriched. And so following that interpretation, we see that the PLS positive gene set, which is associated with the red MCPs, are enriched for, are more greatly expressed in astrocytes, microglia, oligodendrocyte precursors, as well as neurons. And similarly in the PLS negative gene set, which is associated with the blue MCPs, we see that they are more greatly expressed in oligodendrocytes and endothelial cells. And in a similar vein, we can look at biological processes enrichment in addition to cell types. And to do that, we can agglomerate the weights of genes that belong to a specific gene ontology category. So here, those might look like the categories of ion transport or neurogenesis to identify which processes are most highly enriched. And these word clouds show a selection of the most neurobiologically relevant, significantly enriched categories. So broadly, we see that the PLS positive gene set is enriched for genes that seem to be generally associated with signaling between different neural cell types, whereas the PLS negative gene set is more associated with the production of support cells as well as early stages of neurogenesis. And finally, we can examine disease-specific enrichment. And to do that, we can use a database of disease-associated genes to calculate what is the proportion of genes related to a specific disorder, such as schizophrenia, in either the PLS positive or PLS negative gene set, relative to the proportion of disease-related genes in some larger reference set. And we observe for the PLS positive gene set that these genes overlap significantly with genes related to psychiatric disorders, particularly mood disorders such as unipolar depression and major depressive disorder. And schizophrenia was interestingly also among the most enriched or overrepresented disease terms, but didn't pass our FDR threshold. And in the PLS negative gene set, in contrast, we see more enrichment for genes related to intellectual and developmental disabilities. And this pattern kind of lacks the same enrichment for psychiatric terms that we saw with the PLS positive gene set. And so to summarize, we identified that disordered thoughts and sense of self dimension had a macrostructural signature in these data-driven morphometric covariance patterns in the brain, and that these MCPs converge with large-scale cognitive and functional architectures, suggesting a possible structural functional scaffold for psychosis spectrum emergence, and that genes that maximally differentiate psychosis spectrum-related MCPs from those that aren't psychosis spectrum-related might be related to specific cell types, cell signaling related functions, as well as psychiatric disorder related genes and possibly psychotic disorder specific genes. And so broadly, this work suggests that by leveraging patterns of variation across multiple morphometric features, as well as a dimensional characterization of symptoms. We can potentially attain a more nuanced characterization of brain structure in the more normative side of the psychosis spectrum, and potentially identify new vulnerability phenotypes. So thanks so much for listening, and if there are any questions, I'm happy to take them.
Do we have any quick questions? Okay, maybe just one question is, do you know of any evidence for patterns of white matter connectivity that underlie, the connectivity within or between these components that you found?
So that's not something that I've investigated personally, but I know, just thinking of the literature, the morphometric similarity mapping work that I presented early on, which is kind of similar conceptually in that it looks at variation of structural features across the brain. That paper and related work found kind of strong correlations with underlying white matter connectivity. And so I think that could be like a possible future direction. I wouldn't be surprised if they were connected in that way as well. Yeah. Thank you.
Great. Well, thank you so much, Alyssa.
Thank you.
All right. So moving on, you've heard a couple of talks now about mostly about brain structure, although there was functional connectivity. But now we're going to go full tilt into brain function, and we have a talk from Giulia Barachini, who is doing her PhD with Nathan Sprang. So please join me in welcoming Giulia.
Well, thank you so much. I'm very excited to be presenting today some of my work of my PhD looking at brain dynamics on different spatial scales, from brain regions to brain networks. And I really hope to convey the point that we should all be looking at brain dynamics because they're very exciting. So first, before we go into the content of the talk, I would like to first define what I mean by dynamics. And the property of being dynamic is a property of any physical system. And it can be defined as a quantifiable change over time in the output of a system. And in our case, it would be of the brain. And this change could be quantified in order of seconds, minutes, hours, or even years. And we can observe this change also on different spatial scales. In the context of the brain, we can go from microscale, so those would be neurons, to more regions, networks, and functional hierarchies. And we've heard about gradients and hierarchies before, but just to make sure that we're all on the same level, when I talk about hierarchies, I talk about this line of organization of macroscale brain regions and network that separate systems that are more integrated. So those would be lower level systems, such as the visual system, the motor system, from higher order cognitive systems, such as the control network or the default network that are more involved in functions like memory or cognitive control. Now, because I do macroscale cognitive neuroscience, today what we will be looking at and talking about are dynamics in the level of seconds and years. And when I say years, I'm talking about age. And we're going to look at macroscale brain dynamics. So those would be regions, networks, and how these are interacting in terms of where they lie on the hierarchy. Now, why do I care about brain dynamics? Well, we know that our brain is intrinsically variable, and it's also very dynamic. And brain dynamics have been gained a lot of traction, especially in the context of cognitive neuroscience, because this property of the brain to be variable and dynamic, it's the property that allows the brain itself to adapt to the environment. And this is because by being variable, the brain is in this constant probabilistic space. And in doing so, and in being in this probabilistic space, it's able, to respond to any potential incoming information. Now, how do we define macroscale brain dynamics? So first of all, what I use in my research is functional MRI. And I just want to define what we're looking at when we were using MRI. Because we're looking at macro scale function, what we're doing is we're just parcellating the brain in different regions. And so for each region, using MRI, we look at what's called a bold signal. So on the x-axis, we have time. So this would be the time spent in the scanner. And then on the y-axis, we have signal intensity. So that would be the signal that we are observing or measuring from each brain region. Now, knowing this, so that is the output of our system, how do we assess the dynamicity of such output? Well, let's first look at how we are observing regional brain dynamics. One way to do so is by quantifying what's called bold signal variability. And as the term says, what we're looking at is how this bold signal for each region is varying over time. So it's the deviation over time in each regional bold signal. And there are multiple ways we can assess or quantify bold signal variability. And one way to do so, and this is how I've quantified it in my own research, is by using the root mean square successive difference, which is something that has been extensively used to measure heart rate variability, for instance. And so what I'm doing here, I'm just basically taking the first order temporal derivative of each regional time series. And so basically, I'm just looking at how the signal is changing from one time point to the next. And so it's basically allowing me to define a moment-to-moment change over time in each regional bold signal. And so, research using bold signal variability is relatively new. I would say that it's probably only 10 years old in the context of fMRI. And typically, what researchers have done when looking at bold signal variability is relating this metric to a bunch of other things, such as age or behavior. But what hasn't been defined to date is the spatial organization of bold signal variability. And because we've heard in previous talks that space matters because the brain is meaningfully spatially organized, the question that I wanted to ask is, are different regions showing different variability levels? And so what I'm showing here is, I can't really see, how the brain is spatially organized. And so what I'm plotting here on the brain maps is different variability levels. And we have color-coded in different shades of gray, where we have higher gray or lighter gray, indicating higher variability levels. And the first striking thing that you can observe from this brain map is that the visual areas, so at the back of our brain, are the ones that seem to be the most variable. But you can really tell apart what's happening in the middle of the brain. And so to gain a better understanding of the spatial distribution of local or regional variability, I wanted to collapse together regions that belonged to the same functional system or had similar functional properties. And this is basically defined by macroscale functional networks. And so here, what I'm doing is I'm allowing each network to have its own, variability scale. And we're going from, on the left, we have segregated systems, such as the visual system, the motor system. These would lie lower in our hierarchy. And then on the bottom right, we have control systems and default network. And these are higher order networks that integrate information across the brain. And so here, what this result is suggesting is that, space does matter. So variability levels vary by region, and they do so in a spatially meaningful way. And so what I wanted to do next is take the average for each system, so for each network, and look at, how do variability levels actually change based on their functional properties. And so in this bar graph, what we're seeing is, on the y-axis, we have the regional MSST, so that would be the regional variability. And then on the x-axis, we have our networks going from segregated systems, so those would be the visual network, lower attentional network. And then on the right, we have our integration systems, so those would be the control network, the default network. And what we can see from this graph is that the visual system, it's doing its own thing, right? So that's the system that is showing the highest variability. And it kind of makes sense if we think that this is a system that is segregated from the rest of the brain. On the other hand, networks such as the control and the default, so right on this graph, are showing lower levels of variability, which also makes sense because these are networks that tend to form a lot of connections with the rest of the brain. And so if these are the networks that are talking with other regions, being more stable allows them to be more predictable. And so you can see where this is going. So I wanted to then quantify, the association between variability and connectivity that now we're just, observing from a qualitative point of view. And so I took this a step further, and I wanted to actually assess what's the relationship between variability and connectivity. And this has been extensively studied at the microscale level in that, multiple lines of work have shown that neurons that belong to the same, population and tend to form synaptic connections with one another are the ones that also tend to share cortical variability. But this question or this relationship hasn't been really probed at the macroscale level. And before we go into the results, I just want to re-highlight how we calculate connectivity at the macroscale level. So this is purely a statistical construct. We have, again, our time series. We have two regions in this case, time on the x-axis and the intensity of the signal on the y-axis. And what we're doing is we're just collapsing across time. We are taking the Pearson correlation across time between two regions. And so this allows us to come up with what you've seen in previous talks, what we call a connectivity matrix, where we have as rows and columns different regions, and each entry of this matrix indicates how strong the connectivity between two regions is, or is. And so now the question that I wanted to ask is, okay, how can we relate these two things, right? So we have regional variability, and then we have resting state connectivity. And here I highlighted that this type of connectivity is static because, again, we are not really caring about dynamics in this case. We're just collapsing across time. Now, you may wonder, how exactly am I going to relate a measure that comes from one region to something that is in reality relating multiple regions with one another? And that's a great question because I asked that to myself as well. And so what I did next is to try and really probe the relationship, not just in magnitude, but also in topography between variability and connectivity. I coined a new term, and I basically came up with a new metric that it's interregional variability. So what I did was I looked at how similar the variability in one region was from the variability in another region. And this allowed me to attain something similar to what you see here on the right. So instead of having a resting state connectivity matrix, now I have an interregional temporal variability matrix where each entry is indicating how strong the similarity between the variability in two regions is. And this is work that has been published last year in your image, and I invite you to take a look at it because I'm not going to spend time talking about it. Okay, so what basically we've seen in this work is that time matters and space matters. So moment-to-moment changes in the regional Bose signal relate to how brain regions talk to each other, and this is what we've talked about, and this is static resting connectivity, and to also their spatial profiles. Now, because time does matter, this is what I've shown you with regional variability, I wanted to look at how the relationship between variability and connectivity actually occurred when we looked at connectivity also from a dynamic perspective. So instead of collapsing across time, what happens if I look at dynamic connectivity? So let's go back to how we defined brain dynamics, and now look at how we define dynamic connectivity. So there's multiple ways you can do so. One way is using a method that it's called sliding window. So what we're doing here, we are creating windows of equal length that could be 35 seconds, 40 seconds, 60 seconds, and then we're sliding the window across our time series. And for each window, we're calculating connectivity. So again, we're obtaining this matrix of region by region, indicating how strong the connectivity or the connection is between two regions. And so this allows us to come up or create a bunch of different connectivity matrices, right, for each window. And so the question that I wanted to ask here is, how similar or dissimilar are these matrices from one another? Because this would tell me something more about, how the flexibility or the stability of a network is and how it may relate to regional dynamics. So I'm going to take you now on a trip to France to actually assess the similarity and dissimilarity of these connectivity patterns. I've used a method that has been only used to classify, I think, Asian noodles, cheese, wine, and I think that's it, that it's called cove status. So this method was coined by a French scientist. I wasn't surprised knowing the examples that he used. And so I'm just going to establish the method using the example that he came up with, so then we are all on the same level and we can talk about brains again. So code status is something similar to principal component analysis, but it works with multiple data tables, and it's basically leveraging the concept of Euclidean distance. So let's say we have 12 different wines from 3 different regions, and we have 10 different assessors that are asked to evaluate these 12 wines. And we have 2 goals in our analysis. The first one is to characterize how similar or dissimilar these wines are, and this is something we can easily do with PCA. But the second goal, that is where code status comes in, we are looking at discovering whether there is an agreement between the different assessors. So the first goal is very easily achieved. Here we have, typically of, a PCA analysis, we have our 2 factors or 2 components that are maximally explaining the variance in our sample. And here we have our 12 wines, and each wine is representing the average of across all raters. And the space is called the average compromise space. Now, the idea is that 2 wines that are closer to one another were rated more similarly across our 12 raters. On the other hand, wines that are further apart, then they were rated differently. Now, the cool thing about code status is that we can take all our assessors' ratings and project them back onto that compromise space. So again, we have our 12 wines, and now what we can do is we can plot our 12 or 10 raters around that wine mean. And because we're using Euclidean distance, the idea here is that the farther for each wine the point is, so the assessor rating is from the mean, that means that assessor provided a more dissimilar rating, right? On the other hand, the closer the point is, the more similar it is from the mean. Now, let's go back to our brains. So the way we define dynamics, again, is using a sliding window approach. And then I wanted to ask the question, how similar or dissimilar the connectivity patterns for each networks were over time. And so we parcellated our brain. We have our different regions. We basically, for each subject and for each window, now we have a connectivity matrix. We obtain, then, similarly to our wines, our optimum average or compromise space. We compute a singular value decomposition or eigenvalue decomposition on this compromise matrix, and this allows us to obtain, something similar again to our wines. So here, what we're doing is, on the left here, we have our wine example, right, where we had the points were the different wines. In our case, the different points are the different networks, and that would be the center of this hull. And then, which you can't really see, and then the different purple dots around the center are the different windows. Now, what we can do is we can define or calculate the area around that center, right? So the center is the connectivity, the average connectivity profile of a specific network. We said that the different dots are the different windows. And what you can do is basically hypothesize that the bigger the area, the more flexible the network. Because again, we're using Euclidean distance. That means that those connectivity profiles are more dissimilar from one another. On the other hand, a network that has a smaller area of the hull, that means that that network is more stable because the windows are more closer to one another, and so that means that they're more similar. And so for each network now, we have one metric that defines its dynamicity. So again, I wanted to probe the spatial distribution of these areas and of these networks. And so on the Y now, we have network dynamics, and so that would be the area of the hull. And again, what you can see is that there is a separation going from segregation to integration systems, where we have segregation systems, predominantly the visual, again, doing its own thing and showing a high network flexibility. And on the other hand, integration systems, so that would be the control and the default, particularly A and B, are showing a more stable network dynamics. And if you think back of one of the first findings I showed you, this is really nicely recapitulating what I found at the regional level. So again, it's not just that dynamic connectivity levels are varying by network in a spatially meaningful way, but also recapitulating, right, what we saw at the regional level. So in the last bit of this talk, I want to shift gears and look at the impact of age, and this is because I'm coming from an age lab and I'm really interested in age. So the cool finding here is that, again, no matter what spatial skill we're looking at, with age, we see lower brain dynamics. And this is basically shown on the left at the regional level. Those are predominantly lower level regions that are showing an impact of age in that variability. Regional variability is decreasing. But on the right, we also see that this is the case for networks because the area of the hall is decreasing, right, as we go from the top all the way to the right bottom. And the coolest thing is that it's not just that decreased connectivity or brain dynamics, whether it's the regional or the network level, are associated with age, but they're also associated with behavior. And they're associated with trait-like measures of behavior, such as emotion. In fact, they're associated with greater positive emotion and decreased negative emotion. Now, if you have a psychology background, you may think, well, this is really cool because it explains one of the most renowned neurocognitive aging theory, which is positivity bias, right? Because older adults tend to, gear towards more positive, I would say, ratings. So to conclude, I hope that I got you excited about brain dynamics. I've shown you that our brain is intrinsically variable and dynamic, and we should look at brain dynamics on different spatial skills. And that by taking into account brain dynamics in our own research, we can provide a more comprehensive characterization of functional brain organization. And then brain dynamics are cool because they relate to a lot of things such as age and behavior. Whoops. And with that, I would like to thank my supervisor, our lab, and our collaborators.
Thank you so much, Julia. Questions?
No questions.
I'll ask a question.
No questions.
I'll ask a question. How do you think your results would change, or how would they look if you integrated the subcortex or even the cerebellum into the analysis?
Yeah, that's a great question. I think they would, because what the latest research on bull variability has shown is that whatever we see in the cortex is going in the opposite way in the subcortex. So I think I would probably see opposite effects. Yet there's only a few papers looking at this, so I can't really come up with strong opinions. But I'm working on integrating subcortex for sure. Thank you.
Thank you, Julia. Thank you so much, Julia. So you've heard now a few talks about brain structure, brain function. You've seen that the modern IPN neuroscience student is well-versed in a dizzying array of methods. And so for our last talk, we have Justine Hansen, who I'm very fortunate to work with, who will tell us a little bit about how, if you wanted to do some of this stuff, how you would do it and how you would integrate it into your own work. So please join me in welcoming Justine.
Hello. Yes, I'm Justine. I'll get this started. Okay, awesome. So yes, I'm going to be presenting NeuroMaps, which is a software toolbox for contextualizing human brain maps. So this is going to be kind of method-y and like software, and it's kind of going to be like an extended advertisement to try to convince you to use this. Pretty much, we've gotten really good at getting an image of the brain, getting an idea of what the brain looks like, but over the past few decades, with the development of advanced neuroimaging techniques, there has been this explosion of different kinds of brain maps. So maybe not just anatomically what it looks like, but let's say you're interested in like metabolism or a specific feature, specific process. And with all these new brain maps, there's always a new one to explore. So this makes us not unlike early explorers of the Earth. Let's say that if you're an early explorer of the Earth, you might start with the land masses, where are the continents, and maybe also where are the connections between them. So this is not unlike mapping in the brain, where are the brain regions and what are they doing and where are the neural connections between them. And that's great, and that gives us a great foundation. But on top of that, depending on what you're interested in, maybe you're interested in power consumption. So maybe you want to know how much energy is being used in a specific brain region. So this is like metabolism, right? Or maybe you want to know where your cell signal is really good. These are poorly drawn cell towers. That could be like a receptor in a brain region. So where am I going to be listening for a specific neural signal? And maybe you want to know where your text message is going to send really fast, and you're going to depend on really heavily myelinated fibers, right? Like maybe you want to know where myelination is high or low in the brain. And each of these brain maps, these are cool by themselves, and maybe that's all you need for your research. But as we start to lay them on top of each other, we not only get a more complete picture of the brain, but we also start to get kind of opportunities to find relationships between these maps that maybe were previously unknown. So that's what NeuroMaps is trying to do. So NeuroMaps is the software toolbox, and one feature of it is that it has this library or a data set, and it's trying to provide you with a bunch of different brain maps so you can do your own contextualization with either a brain map that's already in NeuroMaps or a brain map that you've developed yourself. So just to give you a few examples of what brain maps are currently available in NeuroMaps, we have metabolism maps. So this is stuff like glucose metabolism, oxygen use, blood flow, blood volume. Microstructural maps like T1, T2 ratio, which is a proxy for myelination or 5G cell signal, and cortical thickness. Functional maps, like Julia was talking about. Cortical expansion maps, so over development and over evolution, our specific brain regions have expanded in different ways, and that's what these maps are telling us. Electrophysiological maps, this is kind of cool because if NeuroMaps lets you do research on a more multimodal scale, so instead of focusing on a specific imaging modality, here I'm jumping into MEG. So these are frequency bands, these are power maps for different frequency bands coming from MEG and also an intrinsic timescale map. And lastly, genomic maps, which can be worked with Abidjan. That's supposed to not say Abigur, that's a cutoff issue, but yes, Abidjan, which is a toolbox that was developed by Ross Markello. And so NeuroMaps can be used in conjunction with that to get brain maps for each for specific genes coming from the Allen Human Brain Atlas. I'm going to devote a slide for the receptor maps that are included because this is something that I worked on at the start of my PhD. So I was collecting a bunch of PET receptor maps across a whole bunch of different receptors in the human brain for a bunch of different receptor systems. And we do make it all open, so if you're interested in knowing a specific receptor distribution or all receptors, not all, but all that I have, the 19 that I collected, then NeuroMaps would be a good way of accessing those. So I'm showing them here on like a parcellated brain surface, but they are available at the volumetric level. And the code I'm showing at the bottom is just an example of how hopefully intuitive or simple it is to get access to this data. So not only getting an access to what is up there, but also just immediately downloading it, immediately fetching it, and getting it onto your local machine. We do have a contribution pipeline, so if there's something you feel is missing or if there's something that you've collected and you think it would add value, you can contribute. Okay, so it's one thing to have all these brain maps, and great, you can look at them and they look very pretty and you can plot them and that's nice. But if you don't know, in neuroimaging, when you get a brain map, it can come in a couple different forms. So this is like if I were to give you an Earth, if I was to give you like a globe and like a 2D flat map on a piece of paper, and then another like set of like Elman cutouts of the brain. I'm sure there's a more technical term for that, but you know what I mean, right? Like, yeah. And it told you to like correlate them to each other, compare them to each other, but these are in totally different spaces. How are you supposed to do that? And the same happens with the brain imaging data. They can be represented in different coordinate systems, and so what NeuroMaps helps with is it helps transform these brain maps between different spaces using robust transformations. So here are the specific spaces that we deal with. Volumetric images, so MNI 152 specifically, and then 3 surface spaces, civet, FSLR, and FS average, and different resolutions, as you can see. And we use registration fusion to go from volumetric to surface, and then something called multimodal surface matching to go from surface to surface. And again, that code snippet on the bottom, it's really as simple as MNI to FS average. You give me your MNI image, I will give you the FS average image, as long as you tell me the resolution that you want it in. We parcelate as well. So what's nice about NeuroMaps is that all this data is represented at the vertex or voxel level, so it's very, very high resolution. But if you prefer working at a parcellated level, the way I was showing those receptor maps, if you have the parcellation that you're interested in, you can give your map to NeuroMaps or use a map already on NeuroMaps and then parcellate it using our parcellation functionalities. Last thing, spatial nulls. So once we have all these brain maps and we can transform them to the spaces of each other, the ultimate goal is that we can compare them to each other. So this is great. Now we're at that spot. We have them in the same space. We can do our correlation or whatever similarity metric we want to do. And what NeuroMaps does on top of that is it gives you the right statistical analyses to be able to assess the significance of that correlation. So specifically, I'm talking about spatial nulls, where you take your empirical data, and instead of having to use this naive model where we're just estimating a distribution or shuffling the brain regions randomly, we're applying something called spin tests, where we take the brain regions and we spin them. So we get kind of a bogus version that still retaining the spatial autocorrelation in the brain, or parametric versions where we're generating a new brain map that estimates the spatial autocorrelation of your data. So this as well, neural maps will generate for you. It can either spin your data or spin your parcels or labels or whatever it is that you want. Okay, so I've kind of gone through like the main things that neural maps does. There's the transformations, the library, and the spatial nulls. So the idea here is that this is kind of like a workflow, if you will. You can have your input data, whether that's coming from your own analyses or coming from neural maps that you fetched. You can transform it to these different spaces and contextualize it against a whole library of other brain maps that are already included in neural maps. And one nice thing about neural maps is because it's open source and kind of developed by the community, it's going to keep growing. And then using these spatial nulls, you get this enrichment analysis. So you get an idea of what your brain map might look like compared to all the other brain maps that are out there in the literature floating around. So let me just give you an example of that in a little bit more detail. These are two different examples. On the top, this is like a self-generated map, so like my map from my research, for example. This is specifically a cortical thinning map to do with schizophrenia. And on the bottom, that's a map already in NeuroMaps, an evolutionary expansion map. So that's telling me which brain regions are expanding from the macaque brain, which is the monkey brain, to the human brain. And in other fields, let's say that you have like a gene list or something, and you want to know what your gene list is enriched in. Bioinformatics has like great tools for that, right? Like you throw a gene list, and it gives you all these biological processes that your genes are related in, and that's awesome. And we kind of wanted to do the same thing for brain maps. So you throw a brain map at it, compare it to a bunch of different the library that's already in your maps, and you get this enrichment. So for example, the schizophrenia map, I'm seeing these correlations, these hits with allometric scaling, which is essentially developmental expansion, which kind of makes sense because schizophrenia is a developmental disorder. And with the evolutionary expansion, I'm seeing multiple red dots, so those are the significant correlations, but one interesting one is maybe individual variability. Do you see my mouse? No, you don't, okay. Individual variability, so brain regions that are more variable in terms of their function in different people tend to be more expanded across phylogeny. Okay, so that's my pitch for NeuroMaps. Do get going, it's up there, so you can pip install NeuroMaps. It's a Python-based software toolbox, open source. If you have a bug, it is relatively new, so it's possible that you run into something. Do let us know. Open an issue on GitHub, we're pretty responsive. Contribute to our open code and data if you realize that there's something that you think could be done better or faster or added to it, just an enhancement. And there's a link right there. Yeah, we have nice documentation, which Laura contributed to a lot, so thanks, Laura. She was the recipient of the oh my gosh, I forgot the second name, the Sievers Foundation. And yeah, that's the link to our preprint, which is going to come out next month in Nature Methods. So thank you for listening, and huge thank you to Ross on the corner there, who is like a lot of the muscle behind this. And of course to our collaborators and contributors who helped kickstart NeuroMaps, but also are helping keep it going, keeping it alive, even though it's like coming out very soon. Thanks.
Great. Thank you so much, Justine. Do we have any questions? I'll ask a question. This is more like our one-on-one meeting. What else should we add to the toolbox, do you think?
Well, give us your brain map. So definitely more brain maps. I think that'll make it awesome. Getting maybe a better view of the whole brain. Right now it's kind of cortex-centric. Not only cortex-centric. We do have volumetric maps of the whole brain. Yeah, and then you can go look at our like issues page on GitHub. Not that there are lots of issues, but there are a couple that we're working on and enhancing. Those are like other things that we're working on right now.
We have
Oh yeah.
Sorry,
Oh
we have
yeah.
one question here and then one question there.
I have one question. May I ask what level of GPU you use in the neural maps? I didn't find that. What level? 2080 or 3080?
No idea.
Okay.
It's not GPU.
It,
I mean, what version of GPU you use? You need requirement in that package.
I don't think we need a higher GPU requirement. No. I mean.
You call it the figures, but you don't need GPU.
No.
We can discuss it.
Yeah,
offline.
I can figure that out, but I don't think so.
We'll
Yeah.
get to you in a second.
Yeah.
Yeah, we have another question there. Yeah.
So you showed that your toolbox can incorporate electrophysiological data.
Step up to the microphone.
Sorry. You
showed that your toolbox can incorporate electrophysiological data. Does it mean like I can record from the brain with like vProbe or like an array and then add this data set to your toolbox?
So not quite. So what we have in the toolbox right now is data that's represented in 4 spaces, so FSLR, FSAverage, CIVET, and M9-152. So if you have a recording and a brain map that is in one of those spaces, like originally, yes, absolutely, we can add that. I mean, theoretically, we can add that, like if it's good and stuff, passes sanity checks. Yeah.
Great, thanks.
Great. All right, so I'd like to, again, thank all of our speakers today, and thank you for your stamina, for sticking around, and we'll see you all tomorrow. I'll hand over to...
Yeah, thanks a lot, Bratislav. That was really interesting, and I think it just highlights the diversity of techniques and skills that we have in the IPN. So right now, there's a coffee break. There's still posters up. I would ask people who have posters, please keep them up until 5:30. I have not had an opportunity to see, I've been running around like a chicken with his head cut off, so I haven't seen many posters, and I hope that your posters will stay up until 5:30, so I'll get a chance to see as many as I can. And we'll see you all tomorrow afterwards. Thanks a lot.